Okay. Okay, fair enough. So let's get started. My name is Stephen Skiena, and this is supposed to be CSE 549, Computational Biology. So I'm hoping everybody's in the right room. Let me start out with reviewing sort of the syllabus and the handout, the lecture schedule before I get into any more details. First thing I'm going to do is pass out a sign-up sheet. I want to make sure everybody signs up their name, their email, what department you're in, and whether you're registered. If for some reason you're not registered, okay, let me know. And if there's anything else you think I really need to know, and you can fit it on the line, tell me. But that's probably enough. Let me start passing this out. Okay. So I'm going to start out with the lecture, going through the lecture schedule and the syllabus. They were here to pass out, but let me pass these around the room, just so everybody, if they didn't pick one up, can get them. There's another set on that side of the room. So pass these around if you haven't gotten one yet. Again, my name is Steve Skiena. If you look at the syllabus for the course, you'll get my number, my office number, my address. I live in the computer science building. I'm there quite a lot from Monday from Tuesdays, typically Tuesdays through Fridays. So you should be able to catch me in general if you wander by. But I'll have office hours from 2:15 to 3:45 on Tuesdays, Thursdays. That's before class. You may want to catch me after class, or make an appointment is also a possibility. The textbook we're going to use in this class is a book by Jones and Pevsner called An Introduction to Bioinformatic Algorithms. And it is a good book because in this class, again, it's a computational biology course. What I think that means is some of you know computation and know biology, and some of you know biology and know computation. How many people here are not CS people? Raise your hand if you're not a CS people. Okay. The non-CS people I treasure in here. Okay. These are the life science people. Typically, some of you are applied math. We're going to count that still as life science for now. So I very much like having the life science people here. I've had several life science people over the years take it who have benefited. What we're going to be talking about in here mostly are about the algorithmic or computational issues in analyzing DNA sequences. That would be the big picture of what we're going to actually do in here. And we're going to talk basically about what the problems are, why they're important biologically, and then what are the techniques that go about solving them. And so the idea is basically you have to understand enough about the algorithmics to try to figure out why these methods work and don't work. That's the useful thing that we're going to be talking about in here. So we're going to go, this book is very good because it both goes into the biology as to why this stuff matters, as well as going through the algorithms on a reasonably accessible level. So I do encourage everybody to look at the book and read it. And if you look at the lecture schedule for the semester, I list under one column, reading. Reading is supposed to refer to what page numbers in the book are we sort of covering in that material. Okay? And so I think it's a good book. I do like it, and I encourage people to go ahead and do that. Any questions about the textbook or anything else we've done so far? There's other books on this list that I recommend for people who are interested in this stuff. Perhaps the one that I would most strongly recommend is there's a book by Gusfield on algorithms on strings and sequences. If you want more details about the real algorithmics underlying this stuff, that's probably the best book on string algorithms, especially as applied to biology. So I would encourage you to look at that one. The grading in this course is going to be done as follows. I am hoping we will have 3 homeworks in here. Last year, we did not have homeworks in the class, and the students all felt that next year's students should have homeworks. It was, I think, a good thing, because I think that there's somewhat of a gap between the lecturing stuff that I'm doing and getting a hang of it. Now, the catch with this is that we need someone to grade the homeworks, and this depends upon me actually getting a TA and stuff like that. So there's still some debate about whether or not we're going to be having homeworks. I'm hoping we're going to have a TA, and I'm hoping we're going to have homeworks. And my resolution may slip if we don't. There's another chunk of the course, close to half the course grade is going to be based on a final project. Okay, I'm going to pass out in about 3 weeks or so a list of possible project topics. And here you're supposed to work, usually in small groups, on some kind of a research-like project related to something that we're going to be covering in here, something related to, computational biology, usually the sequence stuff we're talking about. But I'm pretty flexible about that. But for the life science students, this is an excellent opportunity if you have a project for something that would benefit from having a computer scientist to help work on this thing. I'm very happy to hear people if they've got ideas for projects that they would like to help lead. That would be interesting. I don't want people to sort of say, oh, I'm going to go work on my own research and, let's say, just work on my own project. But if you have an idea for a project where we can incorporate some other students, I think that would be a very exciting thing. So just start to think about that if you're a life science student. We're also going to have a midterm exam in here and a final exam, each of which count for roughly 25% of the grade. And the final exam will be cumulative. Any questions about the grading? A few other details about the grading. I have in past years on the midterms, because sometimes there's a certain algorithmic component there. I have awarded sometimes handicap points to the life science students, okay, if they don't understand the algorithmics properly. And I will continue to do that if that's, if the backgrounds aren't strong enough. Last year I was proud to say the life science people did better than the CS people for the first time. So I gave them some bonus points. But in general, the field I think is getting more level. Largely because I think in the life sciences there has been a growing realization that computing is an important part of what you guys do. And so I think people are coming in with more background than they did 5 years ago, 10 years ago. But anyway, we'll worry about that when the time comes. The other issue about grading is that I will grade on a curve. So if I grade on a curve, typically, half the people will get in the order of A's, half the people will get in the order of B's, and a few people who neglect their business will get below that. So recognize that half of you will not be getting A's in here, okay, at least, okay? And so, it's important to work and it's important to do well relative to the other students. Any questions about the grading or anything like that? About any of the course requirements? Okay, continuing on the syllabus, again, basically we'll be talking about computational biology, which I am referring to sort of problems in genomics, focusing on algorithmic stuff. I'll go through the topics in a few minutes, but we'll be talking about sequence assembly, sequence comparisons for DNA and protein sequences, microarray analysis, protein folding, and phylogenetic trees, and sundry other topics. So it's designed to be a fairly broad survey of what goes on as computational genomics. Any questions? Okay, continuing on the back of the thing, I talked about the homework assignments. I talked about the graduate student project and the exam. Will make available on my webpage an old midterm so people can help study from that. Okay, so if you should take a look at that before you do it. Any questions? Yes?
Will you be doing the same for the final?
No, for the final, I'll figure by that time you figured out sort of roughly what kind of tricks I have. And so I'm not planning on giving you a practice final, but it will be like the midterm, except it will be cumulative. Okay? So that's basically it. Yeah.
How much background in biology is required for this course?
So the background, I don't expect any biology coming in. I mean, to be perfectly honest with you. I do expect a certain willingness to try to do some reading and try to understand things. Again, to a large degree, you can abstract the biology. So it's relatively, it's not that overwhelming to understand what we're actually doing in here. And, so we'll be somewhat gentle there, but I am going to hope people are going to actually do some of the reading. This is one reason why reading the book is a very good idea, okay, is that it will go into give you another perspective on some of these things. And I think that's kind of an important thing. So I'm not scared off. If you're a computer scientist, I am not scared off if you have not had biology. I am a little scared off if you've not had algorithms, okay? So I would like to see my CS people have had algorithms course before, because we are going to go into some details of algorithms, dynamic programming, suffix trees, things like that. For the life science students, I am not worried about them going in, having that great a detail in algorithms, because they will, the life science stuff will be very routine for them. And, I think it's very good for them to, they can benefit a lot from, a gross understanding of this material. So like the life science students have done quite well in here in the past. Okay, and any questions about what background it is? Okay, good. Okay, so what are the rules of the game? Again, this course is going to be basically, I taught this course last year. I'm using, grossly similar lecture notes and stuff like that. Go to my webpage. You can get the lecture notes for the course. The other thing that is going to be good, supposedly, is if everything is right, at this moment, I am being filmed, okay? That there's supposed to be an eye in the sky somewhere in this room. I think it is back there. And I am wearing this microphone not so you can hear me, but so the tape can hear me. And if everything goes according to plans, there will be videos on the web of what goes on in the course. So if you happen to miss a lecture, you can follow on in there. And I encourage people to try it and let me know what their experience is with the tapes, okay? And it's on the solar system for those of you who understand it. The first person that tells me they saw my video will get a brownie point, okay? Any questions about that? Yes?
The videos from 1997 count?
Well, the videos, I have never been videotaped for this biology course. If you really like the videos, you say, I want to see more of your videos. I am videotaped in my algorithms classes and my discrete math classes, and even a computational finance course I taught a long time ago. So if you really decide you can't get enough of me, okay, you can go to my webpage and follow through those things. Yes?
They're still using tape?
Well, they were at some point using tape. These are now being digitized, and so they will go into there will not be a physical tape made of this.
Oh, okay.
Okay, it will be on the web. Okay, I have been taped, and we digitized the tapes. That's what's on the web. So anyway, for this, so we'll see how this goes. I do find it's a useful thing, and I do hope that that this works out. And it may take us 1 or 2 lectures to probably get the recording situation optimized, but that's a good point. Any questions? Okay. What else is there? The other thing that's interesting is that computational biology at Stony Brook is undergoing a big moment now, because there is a wealthy former professor here named Laufer has donated money to start a center on computational biology, the Laufer Center, that in principle is sort of going to really start action about in October 1st. I think that our director, we have a new director for the center who we're very excited about. And so there's going to be a lot of activity on campus related to computational biology now. And so one thing that happens is that they have a computational biology seminar that meets Tuesdays at 4. Now, conveniently, I know that all of you are either available at Tuesdays at 4. Either you're free or you're in this class, okay? So what I might do on certain times is if they have a speaker that is interesting, that is relevant to what we're doing, I am going to encourage us to have class over at the Laufer Center seminar, which is in the I believe they're still going to be in the chemistry building. I'll announce that the first such interesting talk that is likely to happen with the Laufer Center is on the September 21st, okay, where we have Chris Sander from Memorial Sloan Kettering. So that you can get warned that that's probably going to be a day when we're going to be going. And there may be a couple of other ones that are interesting. What else is there? Again, I want people to, if you're a CS student, I want you to have had at least an undergrad algorithms class, okay, before you take this. If not, you shouldn't be taking it. In general, again, there's a lot of other courses at our department. And, you should think about whether this is really one you want to take or not. There's a much larger class than I'm used to dealing with. Last year it was about 40 students. I see we're larger now. Another course that's being offered at this time is Professor Das is teaching a course on mobile computing. And some of you may want to go check that out. And if you want to check it out next time, remember that we are recording the lecture, so you could come back and catch this again next time if you wanted to do that. What else is interesting in here? Again, I strongly encourage the life science people. So if the life science people are getting queasy feelings being here, talk to me before you vanish. Okay? That's what I'd like to say. Go to the course webpage. The course webpage here is www.cs. Actually, I think I can write this down, although it's in the handout. It is my personal webpage, www.cs.sciena/549. And there you'll find the homeworks and the lectures and all the documents and stuff like that. So I encourage people to check that out. Any questions? Other thing, there's two pieces of boilerplate that are important. One is, of course, that students in here are supposed to work honestly, and there's sort of university policies about people who cheat. Which ways can people cheat in this class? Well, one is by turning in a project that you didn't do. That's a good way. Another way might be trying to break into somebody's accounts, trying to steal an exam or something like that. Another might be, having a report and not citing where you get things from and stuff like that. That's plagiarism. I encourage people to act honestly, and it's a dumb thing to cheat. If you do, there will be the university policy there. Any questions about that? And finally, one other interesting, important thing is that there is a Disabled Student Center, okay, on campus. For some of you may have some kind of a condition that affects learning, okay? Maybe you're blind, maybe you're deaf. I once had a student in my class who was deaf, and I didn't learn till the end of the semester, okay? And that, of course, is dumb, because it's helpful for if you've got some kind of a condition. There are, if you're a new student here, perhaps a surprising number of services available. For example, again, I didn't expect this, because you can see we have two people signing, okay, in here. Wave to the crowd, if you can. Okay, good. And this is a sign that there are services available for people in here who need these, okay? And so if you do need this, you should register with the Disabled Services Student Center and let me know about these things. Any questions about that? Okay, that's important. Okay, anything else about the syllabus? Okay, let me do a quick overview of the lecture schedule, just to sort of let you know what we're going to be doing in here for the semester. The first, let's say, 3 classes or so are going to be just basic preliminaries to catch everybody up. I'm hoping today to teach biology to computer scientists. Admittedly, it will take a little more than 1 lecture, so it might bleed onto the second day. We're then going to have 1 day where we teach the relevant computer science to biologists, okay? And again, these are good times to ask questions about very fundamental things that may be bothering you. So some of these questions, this is a good time to ask questions that you might perceive as very elementary or dumb, just to clear them out, because I can guarantee you other people are thinking the same thing. And finally, we'll talk a little bit about what resources and tools are available for computing, okay? And that's something to keep in mind. 2 other things to note on the schedule, at least at the beginning part of the semester. On Friday, we will be having a speaker in computer science from Cold Spring Harbor Lab, which is a very famous biology lab that's located on Long Island, maybe about half an hour, 45 minutes away from us. From a PhD in computer science who just joined there and is working on doing things like DNA sequence assembly and search for new sequencing technologies using modern computing technologies, things like Hadoop, MapReduce, these kinds of processing. So I think that's actually going to be a relevant talk for all people concerned. We're going to be talking in here more about assembly and problems like that. So, but I think it would be good to go to this talk. And the other thing to note is that there is one class we're going to miss the second week. There's no class because of a university holiday. After the preliminaries, we're going to talk for a couple of weeks about DNA sequence assembly. Namely, all of you have probably heard that the human genome was sequenced, or we'll talk about that. That was a big moment in, let's say, biology / computational biology. But what does it mean to sequence a genome, and how do you actually do it? The hard part of sequencing a genome, I will argue, is computer science, and not, it's in some sense very routine factory level biology. And then there is an important algorithmic issue that's necessary to do genome level sequencing. And we'll talk about how that is done, how that was done. And that'll introduce us to algorithms on strings, variety of issues like that. After that, we're going to start talking about homology search, meaning sequence comparison. Probably the biggest thing that biologists do with sequences once they've been obtained and stuck in databases is look, given a particular gene, where are there similar genes? Okay, where are there similar sequences? What do they mean? And that involves a lot of sequence comparison. So we'll talk about algorithms for doing this. Many are going to be based on dynamic programming, on some sophisticated data structures, things like that. And we'll talk about homology. And after that, that's when we're going to have the midterm. Okay? Any questions at that point? After that, we're going to have a unit on gene prediction. So namely, starting to interpret what the data in a genomic sequence is. Okay, that a genome sequence is a string, but there's important features in that string, namely genes, which we'll talk a little bit about today. And we're going to talk about the methods that people use to try to annotate these genome sequences so that they make sense, okay, and to understand what they're doing. And that gets into things like hidden Markov models and things like that, pattern recognition of problems. Afterwards, we're going to talk about a unit on microarray analysis. Microarrays are ways of measuring whether genes are, turned on or turned off, and we'll talk about what that means later on. But they generate amazing amounts of data about the state of biological systems. And analyzing these large data sets is exactly the kind of thing that you need high-power computing for. And so, high-powered statistical techniques. And so I'm going to talk about these array-based arrays, how do they work, what can you do with them, and how do you analyze that kind of data. And hopefully then get a tour of a microarray facility. So we'll actually have hopefully a field trip to a lab here where they do some microarray analysis. So you guys can look at test tubes and things like that and see what they look like. We're probably going to try to have a short unit on proteins, just so people, some issues about protein folding and things like that. Although exactly how much we do in here will probably depend upon whether we've fallen behind or not. And the last unit in here that we'll talk about is going to be on phylogenies, basically trying to reconstruct evolution based on data that you see. So somehow, there is an evolutionary history of life on Earth. And in some sense, we only get data about what species are currently here. And the question of to what extent can you reconstruct evolutionary history, and how do you go about doing that? That's the problem of phylogeny. And we'll talk about some of the algorithmic issues there, because that's another area where algorithmics are very, very important. Any questions? Those are the basic topics that we'll be covering in here. So any questions about any of the course logistics? Yes?
Is this course outline on the website?
This course outline is on the website. So everything should be on the website. If not, you should let me know by email if there is something you think I should be giving you that I am not. Any questions? Okay. Okay, good. So that said, let's start. Let's see if we can make this work. Okay. I'd like to now get into the subject matter for the course. And talk a little bit more about why do we do things, okay? And why is there a computational biology? What is computational biology? And the way I interpret it, computational biology is the application of some core technology of computer science. Some algorithms, is sort of where I come from. So I'm most interested in, combinatorial algorithms. But there are people who work in computational biology who have backgrounds in artificial intelligence or databases, but trying to apply some core technology from computer science to biology. And the reasons why this is an exciting area in my mind for doing, for working is, one, the problems tend to be very, very large. Okay, we're going to be talking, when we talk about the human genome, we are talking about a string that is approximately 3 billion letters long. Okay, a string of 3 billion characters. And, that's big. Okay, and so algorithmics of what you're going to do on a sequence that big. Okay, the algorithmic efficiency matters. And it gets even worse when you start thinking about searching a database. Now, what if you have a database of, hundreds of billions of sequences? How do you search this efficiently? So the problems are big enough that algorithms always, tend to be interesting. The problems are, in general, relatively fresh. This is one thing that's interesting about it from a research point of view. 20 years ago, this didn't exist. Okay, there was not a significant level of interaction between computing and biology. I remember the first time I walked into the life sciences building, I said, oh, I'm a computer scientist. I'm interested in biology. What can we do? How can we interact? They thought I could install Windows on their machine. It wasn't the sense that computation was something that they would do. But that changed very, very quickly. And it is very clear that, people are happy to see me around the life sciences building today. And there's interesting stuff to do. And again, that's because biology is largely is becoming a computing science, especially in certain branches of biology, where a lot of people do their experiments not in a wet lab, but sitting in front of a terminal. And that's kind of an exciting thing. Any questions about that or anything so far? So what other things are interesting? Is this going to be CS or is it biology? In some sense, computational biology has emerged as a mix of this. It's the case that, one thing that's interesting is that there are many problems that were, let me see if I can just do this just to see if I can figure this out. There are many problems that started out of biological interest that, when you explain it to a computer scientist, are natural computing problems, natural algorithmic problems. Okay, and so the problems, once you abstract them enough, clearly become interesting to computer scientists. And sometimes there are ideas from computer science that go directly to biology. So when you talk about microarrays, okay, which is a very important technology these days, that I would argue is really an idea that somehow came out of the computer science people, okay? And somehow people then started, saying, oh yeah, this would be a good thing to build. And people started building arrays and studying what their properties were and things like that. So there's definitely a good mix of interaction between computer science and biology in this field. If you're going to really do research in this field, you're going to ultimately have to learn some serious level biology. In selective areas. You're not going to become a real biology major, but there's going to be certain areas that you're going to need to know something about. And, and also, know something about, advanced computing techniques. Any questions? Okay, let me see if I can progress this. And this is what I made the mistake of. Let's try this. And let's see. Oh, okay. Can I do this now? No. Okay. Let's see if I can. Uh-oh, trouble. Let's see if I can go and advance this now. Okay. Is this possible? No. Sorry about this. Let's see. Hold on a second. This is what the problem I have had with it. Let's try this. Is that it? Boom. Okay, let's try that. Okay, good. So let's just talk a little bit about modern genomics and how the topics that we do in here have really become sort of mainstream tools in the life sciences. So many of you may, I don't know how many of you, again, this time goes, does anybody remember when Sars came out? Sars was this, a few of you. SARS was this virus that came out of nowhere and started killing people, okay? It was in 2003. First, they sort of thought maybe it was the flu, and then, people were concealing how bad it was. But suddenly you had thousands of, as I recall, about 1,000 people who suddenly died mysteriously of this respiratory ailment, and no one knew where it had come from or anything like that. And the question is really, how do you go from a bunch of dead people to actually know what they died from and what was going on? And 20 years ago, it would be a very painstaking, laborious process to try to figure these things out. But it was amazing as I watched this thing play out how quickly quickly people figured out what was going on. So in February, you started getting concrete news reports that there was seemingly something going on. There was a new disease that had broken out. No one had ever heard of it before, and trying to figure out what it was. A month later, people were able to figure out that it was a virus, and they would figure out it was a particular kind of virus. Okay, and the way they did that was using one of these microarrays that we're going to be talking about. Okay, they had designed, this guy DeRizzi had designed a microarray that would be able to tell what kind of analyze DNA samples and figure out what kind of virus something was. Okay, and this was a big surprise. But once they knew that it was a virus, the next job was to sequence it. And a month later, they had the full 30,000 base sequence of what this virus was. And sometimes a complete description of this virus. Okay? And that was announced basically a month later. And then 3 days after that, you could go to the store and buy microarrays specifically designed to analyze SARS. Okay? And now you have a tool that you can tell if you have a patient, did they get a variant of the virus? Is it evolving? Is it changing? Okay? And suddenly you've got new tools to work with this. A month after that, you could open up your Science magazine, okay, which is a very prestigious journal, and you could get a complete analysis of the SARS genome. It would tell you how many genes there were. It could tell you, where did this thing seemingly evolve from? Again, it was seemingly from, was it from a bird virus or a pig virus or what was the gross organization of this virus? What were the proteins in it? How might it work? Okay? People knew that. Okay? By the end of the month, there was a detailed phylogenetic analysis to see where this virus evolved. Okay? How did this evolve? This virus somehow split off from some other virus. How did that happen? Which virus did it split off from? What were the changes involved? This was done a month later. Okay? And so at this point, you pretty much know all that you need to know, to really get down and do an advanced study of this agent. Any questions about it? Okay? So this to me is very inspiring that the tools in our class can do this kinds of things. And again, the genomic technology keeps improving. Another thing that happened about 5 years later is that there's a research group that synthesized the virus from scratch. So let's say that they wanted to study SARS, but they didn't, they couldn't get access to the virus samples. In principle, they could make the virus from scratch using this DNA synthesis technology because, the sequence provides a lot of data, essentially a complete description of the virus. Okay? And now synthetic biology is now an emerging field where, again, there's interesting issues of how do you design sequences that do things. Okay? So there's a lot of different technologies, a lot of different problems, and suddenly now, using the techniques from this class, people are equipped to understand these things much better. Any questions about SARS or anything? Yeah?
So this technique essentially can create a virus that
So essentially what we mean by synthesis here, okay, is that you can basically synthesize a molecule. We'll talk about sequencing is basically reading a molecule. Synthesis is basically writing a molecule to specification. So in principle, you can give it a sequence specified as a pattern of A, C, Gs, and Ts, send it off to a company, with a check, and say, make me this molecule. Okay? And they will come back and make you that molecule to specification. And this opens up all kinds of new and interesting possibilities. So there's a lot of exciting things that can be done with this genomic technology and important things. And the algorithmic issues that we talk about in here play a role in all of this stuff. Any questions about anything we do in here? Okay, good. So what I'd like to start out with is getting a little bit of a culture. So one thing that's interesting about One of the things that makes computational biology very interesting to me is I think it is one of the more successful interdisciplinary areas with computer science. So somehow there's been a discipline of computational biology that has emerged, of people who are half computer scientists, half life scientists, and they can move in each community. And in order to do that, you've got to understand a little bit about what the background of each community is, okay, or what things matter to each community. And there's sort of social phenomena that are quite different when you work in biology and when you work in computer science. And so I'd like to spend a little time talking about this. And if there's any biologists who want to argue with me, I strongly encourage it. But these are my opinions of sort of my observations of difference. Computer scientists can also argue with me, but I think I'm going to beat you. Okay? But I think that, let's just talk about it. So what is the difference between a computer scientist and a biologist? Okay? Well, first of all, let's think what are the same. Okay? One thing that is clear is that there are many different types of biologists. Okay? So, when we say cavalierly here, oh, the biologists want this, that's as silly as saying, oh, the computer scientists want this. Computer scientists, there's people who study algorithms, there's people who do hardware, there's systems people. These people have wildly different interests and backgrounds. And the same is true in the life sciences. If you talk about a laboratory microbiologist, that is wildly different from an ecologist who studies organisms and how they interact in big environments. And that's wildly different from a medical doctor. So when you talk about these things, you've got to recognize that there are communities and they behave somewhat different. But that said, there are certain phenomena that are interesting. One thing that is interesting, that is cultural difference between biology and the life scientists, is that in computer science, everything is true or false. Okay? One is that you say, oh, we work on digital things and everything is binary, so everything is true or false. But more to that point, we construct our own world. Okay? Computer scientists live in a virtual world. They live in the world of their programming languages and of their machines. Everything is, on some level, deterministic or true. Deterministic, I'll wave my hand about that. But there's a certain level of reproducibility and a certain level of, firmness, concreteness that is there. In biology, nothing is ever true or false. And so when I talk to my biologist and I'll say, oh, how does this work? He says, yeah, this gene regulates this protein. Okay? That's good. Now, is there any, there's another way the protein can be regulated. It could also be regulated like this. There's an alternate pathway. And is that true? Well, there's also a third alternate pathway for this thing, but it's less important. And maybe people don't know for sure that it's true. Everything in biology is pretty much messy. I tend to think that there's never anything that's absolutely true or false. Okay? And you've got to live in this kind of a world. Biologists live in a world where they understand complicated, messy things. I mean, the natural world, they study it, not because they created it, but because it's what we live in and it's what the hand they've been dealt. Okay? Computer scientists make their own worlds. Okay? And so there's a difference culturally in how they behave. Does that make sense? Or any questions about that kind of a world? Okay, good. One difference is that biologists tend to be data-driven, where computer scientists are algorithm-driven. Okay? If you talk to a computer scientist, they'll say, well, you say, what is interesting about what you're doing? They'll say, oh, I have a new algorithm for this, or I have a new program for this. When you ask the biologist what's interesting or important about what they're doing, they will have spent many nights late in the lab gathering data. Okay? And data is the world that they revolve around. Okay? They spend, in principle, the biologist spends a lot of time doing these very complicated data set experiments, typically to get a small number of data points at the end. Okay? And this is something they worked hard for and treasure. Okay? Computer scientists tend not to respect data. And let me now give you a proof of this. How many people here have ever done an experiment on an algorithm to see how fast it was in a course or something like that? You did. Okay? What data did you use on your experiments?
Timing and the output.
Where did you get the data from?
By doing many runs.
And what were the inputs to these experiments?
Well, varying different arguments.
Okay, you were changing arguments. Is there somebody else who did experiments in here? Yes? What did you do your experiments? What was the data you fed your computer? What? From the internet. Okay, from the internet is interesting. From the internet may be real data, okay? And so that's actually where sometimes people get it. Yes?
But often you randomly generate.
Randomly generate. That's kind of what I was thinking about. So most people, in the days before there was internet, everybody happily used random numbers programs to generate random data to run through their programs. And to a computer scientist, this makes perfect sense. You want to evaluate the algorithm, make up some random data, and pump it through. To a biologist, this should make no sense. You're going to make up data and learn something from this? Okay? That's a cultural difference here. Any questions about that? Another difference that is kind of interesting is that biologists tend to be obsessed with discovering things, okay? Whereas computer scientists invent things more than they discover things, okay? So if you talk to when I hang around my microbiologists, they tend to be very well read about the literature on this thing. They'll know that, oh, I care about this virus, and they know there's been 7,000 papers written about this virus, and I'll keep up with the latest literature on this thing, because they want to make sure they know exactly what's there, and they want to discover something. You don't get credit for being the second person to discover something. Computer scientists don't tend to discover things. They don't discover principles of nature or something like that. You tend to invent something, okay? You say, oh, I have invented a faster algorithm for X. That is what you're doing. And, or you may say, I have built a system for doing this, okay? And, so there's a cultural difference there. There's a greater level of paranoia among biologists about being scooped, okay? Because being the second person to discover something doesn't do you any good. As a computer scientist, I don't really worry too much when I'm doing research that somebody else is going to beat me to it. Usually I'm not in a race, okay? But in the life sciences, often there's a small number of very important problems. The winner, okay, gets the glory. The loser loses the grant and disappears, okay? So it's a different world there. Any questions? Another difference is that, like I said, biologists, I find, know more than computer scientists. They read more in the journals, okay? They have a large system they're interested in. They have journal clubs. If you talk to the biologists, they'll say, oh, what do you, they often will get together with other students and they'll each read, one of them will present what was the contents of this journal this week, so that everybody's familiar with the latest results. Computer scientists in general don't know how to read. They don't have to read very much to keep up with other things. Okay, so it's back to the inventing thing more than the discovering thing. It's not, again, I don't want to overstate that, but that's sort of, I think, there's definitely a psychology there. Another issue is that biologists are comfortable that data that has errors, okay? So a typical thing, what is a biologist doing? They've sweated in the lab. They're measuring the width of molecules under these very adverse conditions. They know they have slaved for this data. They know this data has a lot of value to them. They also know the measurements are not always very precise, and they have to live with this kind of thing. Now, computer scientists in general are not very comfortable about data errors. If the data is not in the same format, what does your program do? It crashes, okay? And this is, their job is to make the data into the right format. So recognize that we're going to see in this class an interesting phenomena where there's a lot of algorithms we're going to discuss. There's a lot of problems where if we can assume that the data is perfect and doesn't have errors, there are efficient algorithms to solve it optimally. And if there is errors in the data, then the problem becomes NP-complete, okay? And so one thing we as computer scientists can do is we can go to the biologist and say, oh, get all the error out of your data, and then we will talk. The alternative is that we have to learn to live in a world where errors have data has measurement errors and things like that. Any questions, philosophically? Okay. Another difference between computer scientists and biologists is that biologists tend to live in a hierarchy, okay? Much more so than computer scientists do. That typically in a biology lab is organized around a big PI, senior professor person. This professor person will have an army of postdocs. These army of postdocs will talk to the relatively small number of graduate students often, okay, who often do things. These graduate students may tower over the lab assistants or the undergraduates who are doing internships with them. There is a strong hierarchy there where labs have names on them. So when I talk to genetics students, occasionally they come by and say, oh, I want to do a rotation and join the Skiena lab. Okay? That's the way these people would talk. Okay? In computer science, you wouldn't use that kind of a word because it's not sort of organized around a hierarchy of a big professor here. Any questions? Okay? Again, there's a difference between the ideal of what people think of as a biologist or a computer scientist. If you think about, at least at one point, when you said, what was the goal dream of a computer scientist? Somehow the vision was the hacker in the garage, the guy who was going to go off and, who is it that, you would think about, I guess, was it Steve Jobs in the old days or the Facebook guy? The images of this guy going off in his dorm room, building a, $1 billion company, before he graduates. That's the kind of image of what a computer scientist is supposed to do. In a biologist, you're supposed to, become a professor running a big lab. That is sort of the Platonic ideal. Okay, and so life scientists, one interesting property of them is they can get and spend infinitely more research money than CS professors do. So CS professors, we have grants, we have students, we're good scientists. To run a big biology lab, you have to buy equipment, you have to buy reagents, you have to buy hands to do these kinds of things. Okay, it's a very expensive game. Okay, any questions about that? Any questions about the difference between these worlds? Okay, again, I think it is fun to enter a different world. And so if you're a computer scientist, welcome to the biology world. If you're a life scientist, welcome to our world. But recognize that the rules are a little different. Any questions? Okay, I guess one more slide of these things. Another difference between computer scientists and biologists, which is interesting for those especially who are PhD students, is that the research publication systems are wildly different. So in computer science, you typically live in a world of annual conferences. And your goal, if you talk to your advisor, what is he doing? He is trying to get a paper out for the X conference. Okay, and there's deadlines, and every year he's got a deadline, and he's probably got 3 or 4 conferences he aims for every year, and all the students try to get papers done by that deadline. Because computer scientists publish in refereed conferences. To a life science person, or anywhere else in the natural sciences, conferences typically have no value whatsoever. Okay? That scientists publish in journals, and getting things into an important journal is an important thing. Part of the difference here is the difference because, again, between discovering things and inventing things. If you are discovering things, you need to put it in an archival place that everybody sees and that you worry about that stuff. So racing to get into a good journal is an important thing. CS system is different. Not better, not worse, but different. And, and so one interesting thing is how fast life science journals get refereed relative to computer science journals. Historically, if you send a paper to a computer science journal, you will wait a year till you hear anything. Okay? Whereas you send it to a life science journal, you will get a response in 3 weeks if it's a decent journal. Okay? If it's a very good journal, you might get it rejected in 24 hours. Okay? And the difference is that, that they care about their journals more than we care about them. Okay? It's different. Not good nor bad. Anything like that. The other difference, finally, is that computer scientists live in a world where you can always go into industry. And so some of you guys, if you're a PhD student or something like that, if you're ever depressed, you can say, I can always go work for Google. Okay? The biologists typically need to complete 1 or more postdocs before they can get a good position in a faculty or a good position. So it's a different kind of a world there. Any questions? Any questions about the difference between biologists or computer scientists? Anything like that? Okay, good. Okay, so that said, let's now go into some biology for computer scientists, okay? And so for my life science students, you can certainly laugh at anything I say here if it's too simple. If I say anything that bothers you because it's incorrect, please take me to task on it, okay? So I don't have any objections to that. Most of what we're going to be talking about in this class are talking about things that are sequences, usually DNA sequences, sometimes protein sequences. And at the risk of, okay, let's using this thing. A DNA sequence is a string, okay, as we will think of it. It's a string on an four-letter alphabet, okay? The letters of the alphabet are usually written A, C, G, and T, okay? They stand, at least in this class, for A, C, G, and T, okay? So we don't need to worry so much about the names of these things. But the important thing to think about is that a DNA sequence is a molecule, okay? It's made up of chunks. It's a string of these bases. These bases we will call A, C, T, T, G, C, okay? They are, that's basically what a DNA molecule is. It's a string of these smaller molecules called bases, and there's 4 different types of them, okay? Any questions about so far? Okay, one interesting property that's going to be important to us sometimes is that bases want to bind with their complement. So A and T tends to want to be together, and C and G want to be together, okay? And what does that mean? You can think of this molecule as being a flexible thing. Because A wants to bind with T, you might imagine a molecule doing something like this. Suppose this was an A, T, okay, some other bases. This would be an AT. You could imagine the molecule folding into a particular shape, okay, where sort of the attractions between A's and T's, or here you might have a C and a G, govern the shape that it might take on, okay? Any questions about that? Yes?
Why specifically these 4 characters, A, C, G, and T?
Well, they are short for longer chemical names. So, and they're the first letter of the chemical name, okay? I find it easier to remember A, C, T, and G, okay? Any questions? Okay? So, one interesting property of these things is that these bases like to stick to their other one. And the way to think about it is the molecule is this big wiggly thing. Okay, so it's got a certain amount of bending in it. Okay? And one thing it might want to do is to bend, if left to its own devices, it might bend in the way so that complementary letters stick to each other. Yeah?
I might want to correct you. So DNA generally, in the natural form, it has 2 strands. So it's 2 strings. And the reason why we have these complementary bases is if there is A on one of the strings, on the corresponding position on the other string, T would come. And it would be only T and no other.
Right. Okay, so there are different reasons why we have this. So first of all, good and thanks. Okay? Second, sometimes we're going to be talking about single-stranded DNA in this class, and sometimes we're going to be talking about double-stranded DNA. So the reason, let's say, why, if you have to say why does nature have this binding property, it's probably many complicated reasons. The reason you've probably seen on, logos of drug companies is this double helix thing. Okay, you probably are used to the idea that DNA comes as this double helix. Okay, and what does that mean? That means that each one of these single-stranded molecules has basically a pair thing. So if this was ATCG, this would be CGAT. Okay? So the most, let's say, standard way to see this thing, you have this idea that DNA probably comes in this double helix thing. Okay? The interesting point is that if you have one side of this double helix, because the other one is just a complementary basis, you know exactly what the red string is going to be if you know the blue string. Does everybody get that idea? So this is sort of important for sort of reproduction and cell division and things like that, how genetic material is replicated. Okay? So sometimes we're going to be talking about DNA as a double-stranded molecule. But quite often in here, we're going to be talking about it as a single-stranded molecule. Okay? And perhaps nature evolved this chemistry more for, as the double-stranded, the reproduction thing. But a lot of its life cycle, the DNA exists in a single-stranded state, like when you're reading genes, transcribing genes, and things like that. Okay? And for a lot of the technology things we're going to do with DNA, it lives in a single-stranded world. So this looks good on the drug company logo. Usually, in our class, we're going to be thinking about it in its single-stranded state. Okay? So don't expect to see too many double helix squiggles here. Any questions? Okay, good. The human genome can be thought of as a string of approximately 3 billion bases. Okay? Now that's 3 billion bases plus, you could think of it as 6 billion, because when it's in its double-stranded state, there's the one strand and then the replicated strand. So perhaps every nucleus will have 6 billion bases in it. Okay, but let's say 3 billion strands, bases of interesting information. It happens to exist in a certain number of pieces, which are called chromosomes, but we're not going to really worry about that very much. Think of it basically, the way to describe you and to make all your proteins, you are described by a 3 billion base string. Okay, that is your genome. That is your genetic information. Any questions about that? Any other issues come to mind here? Okay. Now, the main purpose of the genome, as far as we're concerned, is going to be to make proteins. And we'll talk more about what proteins are. But the model that I'm going to have, for the most part, is here is your string of length 3 billion. Let's see if I can get this another color. It's not playing. Here you've got 3 billion bases. Certain portions of that sequence describe how to make the components that you're made of, which are chemicals called proteins. Okay? And these are going to be, it turns out proteins are also going to be described as strings. Okay? Only the alphabet size is going to be 20 letters instead of four letters. Okay? So when we deal with strings or DNA, things that deal with strings, with proteins or DNA, algorithms that deal with strings are important things. Any questions about that? Okay. Let me see if I can do this. I think I can do that. Good chunk. Okay, good. Seem to have done that. Let's see if I can move on. Oh, sorry. What is this? Let's try this. Sorry about this. Okay. So what are genes? Okay. We've talked about the basic vision is that we've got a DNA sequence. You're described by a DNA sequence. The important thing is that that the DNA sequence basically describes how to make certain chemical parts that you're made of called proteins. And when we talk here about genes, a gene is a DNA sequence that acts as a description for how you build a protein. Okay? That's what we mean by a gene. And so you can kind of picture that here is your 3 billion base sequence. Okay? Somewhere on it, there is a special coding sequence, which is the gene, that describes how to make a protein molecule. Okay? And there is a very interesting coding scheme that enables you to go from this 4-letter alphabet that DNA is to this 20-letter alphabet that proteins are on. Okay? And we'll talk more about that in detail later, but let me just kind of plant a seed in your head that somehow the purpose of the genomic sequence is to tell you how to make proteins. Proteins are described by genes. In particular, the gene can be interpreted as groups of 3 letters in a row, where each sequence of 3 DNA letters describes how to make a particular one of these 20 letters, these amino acids as we call them. How many possible 3-base sequences are there? Let's think about it. 64. Why are there 64? If we have a 3-base sequence, there's 4 possibilities for the first, 4 possibilities for the second, 4 possibilities for the third. 4 times 4 times 4 is 64. Okay? So what's interesting is nature has built into it a coding table that takes triples of DNA and maps them to protein alphabet symbols. Okay? And thus, a DNA sequence can describe how to make a protein. Any questions about that? Okay, this may be confusing.
How is 64 mapped to 20?
How do we map 64 to 20? Well, the way that it's done is all the 64 things map to one of the 20 things, but it's not a one-to-one mapping. So there might be a particular type of amino acid, let's call it L, okay? And it turns out that there are 6 different triples that might map to an L. There is another amino acid, let's say call it an M. There is one way to map to an M, okay? So each of the 20 sequences has at least one triple standing for it. Some have many triples for it, okay? But in any case, if you want to read what is the protein coded for this, just look at the table. What is an ATG? Oh, an ATG codes for an M. CCA, what does CCA code for? Well, it looks like it codes for a P. Okay? In principle, that would be something you would look up from this table. Okay? And so how built into cellular machinery is this translation table. Any questions about that? Okay? So we will talk more about this, so don't get too overwhelmed by this. But basically there's this world, we live in this world where DNA gets copied into an intermediate form called RNA, which is not very interesting. It's like a Xerox machine, essentially. It just copies the part that you care about. Then there is this interpretation of the RNA molecule in terms of these triples, okay? And that's really how you sort of describe how to make proteins from it. Any questions? Yeah.
Is gene a sequence of triplets?
Right, so a gene would be a sequence of triples.
How many of them?
Well, it depends upon the length of the protein you want to make.
Okay.
So you can imagine that there are big proteins and there are little proteins, okay? But let's say that you want to have a protein. Here's maybe the entire genomic sequence. There's going to be certain parts of it are going to be genes. If this gene has a protein, has M protein letters, the coding for it is going to be 3 times M DNA bases, because each one's going to be, it's going to be a concatenation of triples for now. Okay? Does that clear it up? Any questions about that? Okay? Any questions? Okay, let's do this thing here. Okay, and let's see what we can do here. I think, okay, good. Okay, so the human genome consists of about 25,000 genes, okay? So another way to think about, let's say, what you're like inside, if we want to describe how to make one of you, you are described by a string of about 3 billion bases. There are about 25,000 regions on there that each describe how to make a protein, okay? You may be proud of that, but mice have the same number of genes, basically. So mustard, I believe, has more than you, okay? So different organisms have different numbers of genes, okay? But basically, these are sort of the different protein molecules that you can make, okay, to be made out, you be made out of. Now you're not, it sometimes describes the parts list on a chemical level of how you make a person, okay? Exactly how you rearrange these chemical parts is where the complexity is, okay? And, but at the gene level, we're really just talking about what the parts are. Any questions about that? So the Human Genome Project, which was sort of the, this famous effort here, basically the first job was to sequence the genome of one person, essentially, okay, and read their pattern of A, C, Gs, and Ts, And now from that, you would have gotten a pattern of 3 billion ACGs and Ds. Then we would want to try to understand which part of this was the genes, the things that told how to make the chemicals that you're made out of. We said that that's about 25,000 of them. You said about how long are they? Let's say a typical protein is about 1,000 bases long, or 1,000 triples. If that was true, how much of the genome is interesting or makes genes? Okay, we said that there would be about 25,000 genes. Each of these would be about 3,000 letters long, right? That would mean about 75 million bases code for genes. And that means that the rest of it, or about 98% of the genome, does something different. Okay? And what's interesting is for a long time, people didn't know what it did. They used to call it junk. Okay? But as time goes by, they find more and more reasons for why this other stuff exists. In general, we're not going to spend that much time talking about the rest of it. But a way to think about it is you are a genome, you've got 3 billion bases, about 25,000 strings of it, subhunks of it, describe how to make proteins, okay? This represents about 2% of the total, but it's important. It describes how to make everything that you're made out of. Any questions about this picture before we move on? Okay? And exactly what the rest of it does is very interesting, but for now, let's not go through that. Okay? Boom. And let's try to go down. Okay. Any other questions about genes or how they are? Yes?
So all of our cells are made up of the DNA? Is that what that is?
So cells, we're going to talk, I think, well, here I guess we're going to start to talk about cells, right? So here, if we look at this thing, cells. Cells are what you are made up of cells, right? Cells are made up of protein and water and all kinds of other things, okay? And they're complicated. We're not going to talk too much here about cells, okay? But what's important is that the program to make a cell is basically governed by the genomic DNA, okay? So a way to think about it may be you are made up of cells, okay? The cells have a membrane. They have stuff inside. We'll call it gook. It has a nucleus inside that would have what the genomic DNA is. Okay? And this describes how to make the cell, that genomic DNA. Okay? And when it goes through a division step, basically it goes through a complicated dance that splits the cell and creates two cells, each of which have the same genetic information, right? So although you're made up of you're made up of cells, and cells are interesting. Cells are made out of proteins. The information content is this DNA sequence. That's why we want to study it, because that describes really all that you need to make one of these things, okay, in principle. Any questions? Okay. Any questions about that? Okay. So one thing that molecular biology is this study of how all this DNA and proteins are made, okay, really on the level of molecules and how this stuff works on a molecular level. And that's very, very important to understanding how life works. Ultimately, though, life somehow feels more than chemistry, okay? And so when we think about life, we usually think about organisms, okay? Humans are organisms, okay? Viruses are organisms in some sense. And so one thing to know is that life gets broken down into different types. There's different types of living things. And it's important to understand the scale of different kinds of living things. Okay? So the simplest things that you might call living, okay, are viruses. You guys as computer scientists, you're familiar with viruses because you get them by email, right? Well, what is an email, if you think about it that way? Why is that actually? It's actually a pretty good term. A virus is a piece of code, okay, that runs on your operating system and causes you problems, right? So that is largely describes what a living virus is. A virus is a piece of code, a short piece of DNA with a small number of genes, okay, that has the property that it turns the cell into doing things that are good for the virus and not for you, okay, in principle. That's basically what it is. So the simplest living things are viruses. And viruses are generally described by DNA sequences of about 10,000 bases, okay? So viruses are really string. You can completely describe a virus. It's about a string of about 10,000 letters long. Any questions? The next simplest thing that exists are things called prokaryotes. These are basically bacteria, okay? And bacteria typically have a genome of about 1 million letters long, okay? So bacteria are much more complicated generally than viruses, okay? By a factor, is that a factor of 100, I think? Yeah, okay. So bacteria are free-living things. They're about the simplest free-living thing you can have, okay? And they're very, very important. More complicated than bacteria are things called eukaryotes. You are a eukaryote, if it makes you feel better, okay? It basically means something that has cells with internal structures in it, like this nucleus that we talk about, okay? So yeast is probably one of the simpler eukaryotes that people study a lot here, okay? At Stony Brook, we happen to have a lot of yeast people, okay, here. And, they are single-celled organisms, okay? But they have cells that are a lot like us. So that's one reason why people would study yeast. They're very simple, but they have cells with the same kind of structures that we do, okay? Any questions about that? And then growing in complexity, you then start to have multi-celled organisms like us. If you think about it, we are very complicated, not only because we have cells that have nucleuses, but we have lots of cells of lots of different types that all seem to communicate, and how that develops, all very complicated, okay? So they recognize that organisms happen at different scales, and the biology happens at different scales. Any questions about that? Yes?
Do all the different kinds of cells have the same 3 billion character sequence?
So actually the answer, do all cells in your body have the same sequence? The answer basically is yes, actually, okay? So you have a brain cell is not that any more complicated than a bone cell or a skin cell from a DNA level, okay? It turns out that what makes these cells different is a question of which proteins are being made at that given time. You can think of the DNA as a description of how you make all the proteins. Something's got to actually make them, you've got to say use it. Okay, you can think of the genomic DNA sequence as like a recipe book for making any one of the proteins. There has to be some mechanism to turn on and turn off proteins. Say, basically, in this cell, I want to make this protein. In another cell, I may not want to make it, okay, unless I have a certain kind of stimulus, okay? So when we talk about microarrays and things like that, we're going to be talking about starting to study what genes are turned on and turned off at what situation. That's very important, because that's the difference between different cell types. It's not the difference that the genomic data is different. It's how much it's being interpreted. When I was young, there was a movie, Woody Allen had a movie called Sleeper. I don't know if anyone's ever seen this. But somehow there was this notion that the person, they had stolen the skin cell from the guy's nose. And from the guy's nose, they were able to reconstruct the person. Because in principle, that's not a scientific thing, but in principle, that one cell would have enough information to reconstruct the whole thing. That's why when you hear about stem cells, there's a lot of stuff in the news about stem cells. That's because those are cells that have the same information, but more than that can be manipulated to turn into any kind of cell by turning on and off the right things. Okay? Any questions about that? So cells and organisms are important. Okay? Any questions? What I think is going to be the last slide I'll talk about today. One last thing about organisms, since we're running out of time. One thing to know about organisms. We agree that organisms are of different complexities. Different people study different organisms for different reasons. Okay? So I heard some snickering when I said, oh, we have a lot of yeast people at Stony Brook. Well, yeast is a good thing, okay? Yeast is a model organism. What is a model organism? Okay? Historically, biologists were organized to a certain extent around what organism they worked on. Okay? So how are computer scientists organized? Okay? You would say, oh, Skiena, he's an algorithms person. Zadok, he's a systems guy. Right? To a certain extent, biologists were historically organized by what organism they worked on. Okay? Oh, he's a yeast guy. Oh, he's a zebrafish guy. Now, why is that? Okay? The reason is that there are different aspects to biology that someone might want to study. And in certain organisms are easier to study certain phenomena than other ones. Okay? So, E. coli is a bacteria. Yeast is a simple eukaryote. Drosophila is a fruit fly. A mouse, you know what mice are, right? These are all famous model organisms, meaning that there are lots of people who study these types of organisms. And out of all the species in the world, people study basically very few of them carefully. Why is it that there are mouse people but not lion people? Okay, can anyone think about this? Why aren't lions a good model organism? Does anyone want to guess? Yes? There are few lions in the world, so it's hard to get lions, right? It's pretty